If you counted every single species of reptile, amphibian, bird, and mammal in the world, it would total only about 10% of the number of species of plants in the world. There are around 400,000 plant species on Earth, with 2,000 new plant species being described every year. So how do scientists track this huge amount of biodiversity? Herbariums, a strange word if you've never heard of it, originally coming from herba, meaning plant or grass, and area, meaning pertaining to. So you might think this room is just a bunch of cabinets with old books, but it's actually quite the opposite. See, in the latter half of the 15th century, a French botanist, Joseph Piton de Tonnefort, switched the definition of herbarium to a collection of dried and pressed plants, and later, Carolus Linnaeus, the founder of modern scientific naming and categorization, streamlined it. In every cabinet, there are hundreds of dried and pressed plants collected from around the world spanning many centuries. This is how scientists catalog plants. So let's talk about how I use herbariums in my own research. I study this beautiful desert-like ecosystem in central Florida called Florida scrub. This ecosystem sand comes from the quartz of the Appalachian mountain range, and its distinct look is a direct result of millions of years of sea level rise and fall. This is why Florida scrub harbors dozens of plants and animals found nowhere else on Earth. However, Florida scrub was almost wiped off the planet. Land use change due to agriculture, residential development, and sand mining almost destroyed this ecosystem entirely. This is problematic not only because of the biodiversity this ecosystem harbors, but also because it plays a major role in water entry into the aquifers that Floridians use for 90% of their drinking water. My research focuses on a group of short shrubs in Florida scrub called pinweed that are threatened or extinct in the state of Florida. One of my main questions about pinweed is how it has responded to climate change and this destructive land use change. I take pinweed seeds from herbarium records collected before the historic destruction of the Florida scrub and germinate them. I can then extract the newly grown plant's DNA to compare to the current DNA of pinweeds in Florida scrub. In this way, I have genetic information from the same location, but before and after the destruction of this ecosystem. Through different analyses, I can start to unravel how pinweed has evolutionarily responded, or not responded, to this climate change and destructive land use change. This can inform conservationists and resource managers on what steps to do next. So while herbarium specimens might not look like much more than old plants in cabinets, they're actually time capsules. Because as our world continues to change, these collections of the past may be our most valuable tool to protect the biodiversity of the future.